You know, in the world of medicine, some of the most dangerous bad guys are completely invisible. Imagine a patient fighting for every single breath because of pneumonia. A silent killer is at work. But how do you catch a suspect you can't even see? Well, today we're going inside the lab for the ultimate forensic investigation to unmask a microscopic killer.
So let's set the scene. The crime scene, a patient's lungs. The crime, pneumonia. But see, that's just a label for what happened. To actually solve this case and, save the patient, we need to find out who did it. Was it a virus, maybe a fungus, or could it be our prime suspect, a very specific and very nasty type of bacteria?
And here it is. Meet our main person of interest, Streptococcus pneumoniae, or pneumococcus for short. This little guy is the #1 cause of something called lobar pneumonia, which is a really serious infection that can take over an entire section, or lobe, of the lungs. So our whole job here is to prove, beyond a shadow of a doubt, that this is our culprit.
All right, every good investigation needs a solid plan. Here's ours. First, we're going to profile our formidable foe. Then, we'll see how the evidence gets collected and analyzed for those first big clues. After that, we'll watch how lab techs actually grow the culprit, make the final positive ID, and then, the best part, choose the right weapon to take it down.
Okay, first things first. We need to know who we're even looking for. It's time to build a profile of our pathogen. What makes it tick? What are its tells? What makes it so dangerous? And you know what's wild? This thing has a rap sheet, just like a criminal. Our suspect usually works in pairs. We call them diplococci. And they've got this unique flame-shaped look. Its identity is gram-positive, which is a key classification that helps us narrow things down in the lab. We'll get to that. It also wears this clever disguise, a protective capsule that kind of looks like a halo under the microscope. And its MO? It causes something called alpha hemolysis, which is just a sciencey way of saying it partially destroys red blood cells wherever it goes.
All right, with a profile in hand, it's time to head to the crime scene. And let me tell you, the quality of our entire investigation hinges on the quality of the evidence we can collect.
So where do the lab detectives look for evidence? Well, the main source for pneumonia is going to be sputum. But if the infection is broken out and invaded the bloodstream, a blood sample is absolutely crucial. Sometimes they might even collect fluid from around the lungs, or, and this is serious, from the spinal cord if they're worried the infection is spread to the brain and cause meningitis.
Evidence secured. Now it's time for the first round of analysis back at the lab. These quick and dirty tests are going to give us our first big leads in this case.
So, the very first stop for pretty much any bacterial investigation is a classic technique called the gram stain. You just smear a sample on a slide, treat it with a few different dyes, and then pop it under the microscope. And believe it or not, this technique is over 100 years old, but it's still an absolute powerhouse for splitting bacteria into two major groups, gram-positive or gram-negative.
And boom, this is our first big break in the case. The stain reveals these purple, flame-shaped bacteria working in pairs, an exact match to our profile. You can even see that clear little halo around them. That's the capsule. This is a positive sighting. It's a huge clue that we found our suspect, pneumonia.
But look, a sighting isn't enough to get a conviction. We need more. In the lab world, that means getting a pure culture. We've got to grow this suspect, force it out of hiding and into the open, where we can run a lot more tests on it.
Now, the big challenge here is that this culprit is picky. In the lab world, they call this fastidious, which is just a fancy way of saying it's a picky eater. It won't grow on any old standard lab food. Nope. It needs a special enriched home, like blood agar, and a custom atmosphere with extra CO2 to really coax it into growing. And when it does decide to grow, it leaves behind a very specific signature. On blood agar, the colonies create that greenish zone. That's the alpha hemolysis we talked about. But as they get older, the middle of the colony actually collapses, giving it this really unique draftsman or checker piece look. And on another plate, called chocolate agar, it creates this distinct greenish, kind of bleached out effect. These are more key clues for our case file.
Okay, so the evidence is definitely mounting. We've got a gram-positive diplococcus, it looks right, it grows in a really characteristic way, but now comes the most important part of the whole investigation, making the final positive ID and ruling out any imposters. Because here's the twist. There's this whole other family of bacteria called viridin streptococci. They're also gram-positive, they're also alpha-hemolytic, and they live totally harmlessly in all of our mouths. They're a classic look-alike that could easily contaminate a sample and throw the whole investigation off track. So how in the world do you tell the killer apart from its harmless cousin?
Well, you do a side-by-side comparison, a tail of the tape. As you can see, our suspect is flame-shaped and has that capsule. The imposter is just round, no capsule. But the real differences show up with two key chemical tests. First, bile solubility. When you add bile, our suspect, S. pneumonia, literally dissolves. The cloudy liquid just turns clear. The imposter stays cloudy. But the real game changer, the final test, is optokine sensitivity. The optogen test. This is our smoking gun. It's a super simple test. You just put a tiny paper disc with the chemical optogen on a plate where the bacteria are growing. And this one little test is going to give us the final definitive answer we need.
And there it is. If the bacteria really are S. pneumonia, they're sensitive to that chemical, so they won't grow anywhere near the disc. They create this clear zone of inhibition. And if that zone measures 14 millimeters or more, that's it. It's a positive ID. Case closed. We have caught our killer.
But identifying the bad guy's only half the battle, right? Now that we know who it is, we need to find its weakness. We've got to choose the right weapon to eliminate the threat and save the patient.
So this last stage is called antimicrobial susceptibility testing, or AST. Basically, we take our captured culprit and we expose it to a whole panel of different antibiotics to see which ones actually work. It's pretty much an interrogation to find its vulnerabilities. And this final step is so critical. It makes sure the doctor picks the right antibiotic, which prevents the treatment from failing. And just as important, it helps us fight the huge global crisis of antibiotic resistance by not using drugs that don't work. It is the final piece of intel that turns this whole lab investigation into a life-saving mission.
You know, when you put it all together, you realize this isn't just a bunch of boring lab tests. From collecting the first piece of evidence to finding that smoking gun, the entire process is a high-stakes, desperate race against time to unmask a microscopic killer and find its weakness before it's too late.
And that really leaves us with a huge question for the future, doesn't it? Our suspects, these bacteria, they are constantly evolving. They're developing new disguises, new ways to resist our best weapons. So the question is, how will our detective methods have to adapt to stay one step ahead in this never-ending microscopic manhunt?